a very special seminar, and I'm very happy to introduce Kathryn. She was an undergraduate student, a math student at Cambridge University, where she was also a master's student. She has got a PhD from Oxford. She then worked at Ocado Technology before joining DeepMind, where she was part of the team that developed AlphaFold, which makes prediction for the protein's 3D structure based on its amino acid sequence. This is known as the protein folding problem. Because of her work, she was listed in MIT 35 Innovators Under 35. Thanks a lot, Kathryn, for accepting our invitation, and please, the word is yours.
All right, thank you very much. Hi everyone. Thanks very much for inviting me to speak here today. Really excited about that. As you said, my name is Kathryn Tunyasuvunakool, and I work at DeepMind, which is an AI research institute based in the UK here in London. I should say, by the way, I must be the only person post-pandemic who isn't an accomplished Zoom user. So if there are any technical hitches, do just let me know and bear with me if you can't see the slides.
Okay.
All right. So DeepMind has been carrying out AI research for quite a few years now. And at first, it was a company that was better known for its work in games. So for instance, training a single agent that can play multiple very different Atari video games. Or an agent that can beat human champions at games like chess and Go. Probably AlphaGo was DeepMind's most famous work for a very long time. They were the first to train an agent that could beat the leading human player in Korea, and there were some live matches that were quite widely reported. Longer term, though, it's always been our hope that machine learning could be productively applied to science. And that's an area that we've progressively focused on. First and foremost, just because it's a very practical application, something that has the potential to actually benefit humanity, which obviously is something we would all hope for from our work. But it's also a really challenging domain, arguably more so than games, because in science, we're training models to perform tasks that no human knows how to do yet. In a sense, it's going beyond what human intelligences have so far accomplished.
I'm sorry to just interrupt you for a second, just to make sure. Are you sharing? Are you showing your slides at this moment, or
should be?
Okay. Actually, I don't see the slides, so.
Okay, no problem.
Did you share?
Do you want me to try and reshare?
I'm sorry, Thiago, do you see the slides, or it's only me?
No.
Okay, so yeah, try to share it again. Well, actually, last time, okay, yeah, now we can see it. Almost. Now this black screen, I mean, you started sharing, but the screen is black for me.
I can see these slides.
Oh. Okay, yeah, I can see it now too.
No, it's good. Okay. So it's working now. But I think Kathryn, we are mute. Please unmute you. We cannot hear you because of your mic is okay. Now it's working.
So I can only speak when I am not sharing my slides, it would seem. As soon as I share them, my speaking is grayed out and it puts me on mute. Do you know anything about that as a technical problem? I can try one more time.
Okay, let's try maybe one more time.
Hi, is that working now?
Yeah, I think now.
Great. I think I know what the problem was anyway, but yeah, if that's all good, I'll continue.
I'm not seeing any video, any screen yet. It's black.
Yeah, I think it's loading something.
Yeah, last time it was like this, so I haven't.
Me as well. It's just a black screen.
How is this? Is that better?
Not yet.
Okay, it says that I'm screen sharing now.
I can't see anything.
It appears like sharing, but the screen is black.
Okay. Let me try a different option. I mean, the nuclear option is I just talk at you guys, which I'm willing to do. It would be a real shame, but.
But we do want to see your slides.
Yeah, I never encountered this problem before. I wonder what it is.
Okay, no problem. What if I were to share my slides to one of you? Would that work?
Yeah,
Yes.
yeah, it might work.
Or someone else to present if we can't get this to work?
Right.
Okay, just 1 second. So nothing's sharing right now, yeah?
No.
You can send maybe to me by email and then I.
Yeah, 1 second. Okay, I am sending my slides to Tiago, if that works.
Okay, for people who just happen to follow us on YouTube, we just have small technical problem here, but hopefully we'll resume soon. Well, incredibly, last time we also had a technical problem with Zoom, and it took us about 10 minutes to solve it. So it was.
Oh, well, at least we're experienced in this, eh?
Well, you know, we thought we were, but the person who gave the talk actually gave many talks before and never had the same problem.
Mm.
Okay, let me. Right. Metal. Then I can start. Screen.
Okay, it's now a challenge for you, Tiago. Can you share your screen?
Just thought it was worth a try.
It's me asking permission.
Yeah, the host has to.
Yeah, Tiago is a host.
Oh, okay.
Now?
Yeah.
Ah, excellent. There you go.
Okay. Okay, let
Thank
me
you very much.
put the full screen. The full screen. Is it okay?
Yep, great. So if you just go on slide 2 and I'll say next slide.
Okay.
Great. Yeah, so as I was saying, I'm here to talk about a particular example of AI for science today, AI for biology, and in particular, training a model to predict the 3D structure of proteins. So over the next couple of slides, I'll try to introduce that problem a bit and explain why it is that we care so much about it. Next slide, please. So proteins are very important molecules in biology because they are basically the workhorses of the cell. Just about any important function that you can think of, there are at least half a dozen proteins involved in that. So they make up the cell structures, like collagen there in the top left. They can form molecular motors, like dynein, for generating movement. They also form enzymes that break down chemicals into useful components. So the example there is amylase, which breaks down starch into sugars. And they can transmit signals into the cell and so on. And proteins are made out of these chains of building blocks called amino acids, which come in 20 different varieties, each of them being assigned a particular one-letter code. Now, typically, these linear chains of amino acids don't stay as linear chains. They rather spontaneously fold up into complex 3D structures, like the things that you see in red on the left. And that 3D structure is then suited for the particular job that the protein needs to do. This whole folding process is driven by physical laws, the properties of the individual amino acids, their particular sequence, and how they interact with each other in the environment. Next slide, please. So there are several reasons why we might want to know about a protein's 3D structure. A very practical reason is that it's useful for intervening in how a protein works. So an awful lot of small molecule drugs, for instance, work by binding to a particular protein and disrupting its function in some way. And if you are aiming to develop such a drug, it might be useful to at least understand vaguely how that protein looks and be able to visualize how a drug might fit inside. For other biologists, even if you're not, involved in pharmaceutical development or something like that, just seeing a structure of a protein can help to inspire new hypotheses and give you ideas about how to plan the experiments that you want to do. But for me, the highly impractical reason to be so interested in protein structures is just that they're really beautiful and fascinating. So it's amazing to me that we can understand down to these molecular details how biological processes work. If you imagine that our knowledge of something like ATP generation stopped with a pathway diagram, it just said protein A performs this function, protein B does something else, just a little chain of boxes connected by arrows. If that were all we knew and we glossed over the details of, oh, there's this big structure that acts like a motor and it's powered by a proton gradient and rotates and so on, I think our knowledge of biology would be so much the poorer if we didn't know all of these details. So that's my highly impractical reason. Next slide, please. So experimental methods for determining a protein structure have existed going all the way back to the 1950s or so. In the beginning, much of this work was done with X-ray crystallography, but now frequently other methods, IOEM or nuclear magnetic resonance, are used. But like most experimental work, it's very difficult and there are pitfalls. So some proteins are much more difficult to work with than others. And there can also be difficulties, for instance, in making enough of the protein just to be able to begin your experiments. There can be difficulties growing crystals for crystallography, or trying to get a clear cryo-EM picture of the protein due to its intrinsic flexibility. If one domain is moving, it tends to be sort of blurred out. And so these are all problems you can have with individual targets. And that means that sometimes these experimental approaches can be quite time-consuming. In principle, though, and with a few caveats, just the amino acid sequence of the protein should completely determine the structure that it ends up in. This was the hypothesis of Anfinsen. And so it should be possible to come up with a computational method that predicts the protein's structure directly from its sequence. Now, while the output that you get is only a prediction, that can still be extremely useful, because we're talking about being able to provide some sort of actionable information about the protein structure in a matter of, say, a couple of minutes to an hour, rather than it taking months or years. The other advantage with computational methods is that you can, in principle, produce structures for all of the protein sequences that we know about, rather than restricting this sort of experiment to just targets that are of really particular interest. Next slide, please.
Just one question, please.
Yeah.
This, in principle, this sequence determines the structure was something experimental, or what is it? You mentioned that there is some caveats and.
Sure. So this idea originally comes from work by Anfinson, where he would denature proteins, basically heat them up or do something else to them to make them unfold. And then he showed that they fold back into the same sort of structure that is still able to do its job. So that suggests that, you know, there's nothing else that you need around in order to make this folding happen. It's something spontaneous. Now, I said that there are some caveats because you do get specialized proteins in the cell called chaperones, and one of their jobs is to help other proteins fold. So they sort of like will bind onto them and sort of, you know, stabilize particular conformations and things like that. So it's not true that it's always as simple as that. Nothing in biology is ever simple. But yeah, in general, we say that the sequence determines the structure.
Okay, thanks.
Cool. Next slide, please. Okay, so that kind of defines the structure prediction problem. And over the years, as various people developed methods to try and solve it, there became a need to really evaluate those methods and figure out how accurate they might be. So to that end, John Molt and colleagues set up a community assessment, which is called CASP, back in 1994. And that's basically run every 2 years ever since. And in CASP, the entrants are challenged to predict dozens of protein structures that have kind of just been solved experimentally, but the coordinates haven't yet been made publicly available in the standard databases, so in the protein data bank. And the nice thing about this, you know, from an ML assessment kind of perspective, is that it's a blind assessment. There's no way that you can accidentally leak data or be cheating or, you know, be using the coordinates in some, you know, accidental way. It's a really good test of our methods. Now, CASP's main accuracy metric has for quite some time been GDT, which you can see in the graph there along the bottom. That's a score out of 100, which you can maybe think of as something like the percentage of residues that are in the right position. And by the way, don't be fooled by this graph into thinking that the field made no progress over this period. It's actually that crystallographers were just solving much more difficult targets. So, you know, over time, the sorts of things that you're challenged to predict in CASP have also gotten more and more difficult. One thing we always like to emphasize is that having this independent assessment has been a huge boost to the field because it helps to provide a clear definition of the problem. And it also gives us a really obvious way to measure progress. Next slide, please. So that was the state of things 5 or 6 years ago, when a few people at DeepMind first realized that, hey, protein structure prediction seems like a really interesting application for machine learning. This was before my time. I actually wasn't at the company back then, but initially it started with a 2-week hackathon. Which just goes to show that nice things can come out of hackathons from time to time. And that later turned into a proper team that got more and more serious. And they ended up taking part in the CASP 13 assessment back in 2018, it would have been. And then in CASP 14, they took part in 2020 with a model called AlphaFold, which is the main thing that I'm here to talk about. Next slide, please. So the reason that AlphaFold has generated so much attention since largely because of its results in this independent CASP assessment. You can see on the right of the slide a couple of the proteins that are taken from CASP 14. The real experimental structure is in green and the AlphaFold prediction is in blue. It's not really quite close. And overall, AlphaFold reached a median accuracy above 90 GDT on the hardest category of targets. That's a score of 90 is something that the organizers described as competitive with experiment, which I think is just about the most flattering thing that you can say about any computational method. So that was obviously really exciting for us. Not every target was predicted to that level of accuracy, but enough of them were that some people have since declared the protein structure prediction problem in this form as largely solved, which I think was beyond our wildest expectations for this project. So that's really what the situation was in CASP. And we also made some progress in capturing the fine details of structures, like the side chains that you can see drawn as sticks in the structure on the top right. And the model was able to handle large proteins reasonably well. Previously, something like the structure on the bottom right would have been chopped up into individual sort of rigid bodies called domains, and then you'd have been assessed on each of those. But actually, you can feed the entire sequence for this protein into AlphaFold, and it does a reasonable job of packing all of the domains together and figuring out what the whole chain looks like. So those were some interesting new developments. Next slide, please.
So maybe a quick question. So,
Absolutely.
so you explained in the beginning that there's the same result in the previous CASPs is not actually a lack of progress, but rather more structures determined experimentally. So, can it be that the opposite, exactly the opposite, happened in the last two? That at some point the number of structures should come to saturation. You cannot continue infinitely producing a structure. So what's the effect, I mean, compared to previous years?
Yeah, so that's a really good question. I guess what I was alluding to earlier is the fact that experimentalists are always pushing the boundaries, and they're able to solve larger and more complex protein structures. And so the targets that you're tasked to predict in CASP get harder. But I think what you're also suggesting is the data set that you have for machine learning is growing all the time. So it could be that we were just able to solve this problem because we got to saturation point where there were enough structures in the training data that we could solve this problem. So obviously it helps to have more data, but an interesting thing is that between CASP 13 and CASP 14, we actually didn't update our training set. So the purple bar and the blue bar were trained on the exact same set of data. There was actually a solid reason why we did that, which is some types of proteins are just quite rarely solved. And if you get a question like, oh, can you tell me how well AlphaFold does on this rare type of membrane protein? It's useful to have several years of test data that the model hasn't been trained on. You can then look through and say, oh, yes, we have 10 examples of those, and I'll go and evaluate the performance and see how it does. So yeah, I think it's probably a contributing factor, but at least between those two time points, the data set didn't change.
Thanks.
Great. So I'm more on the biology side of the team rather than the machine learning side, but I will give you a sort of quick overview of how the network works over the next couple of slides. This is focusing on what happens at inference time. I can take some questions on the training process later if that would help. So the AlphaFold model basically takes 3 inputs. First of all, we have the amino acid sequence that we've discussed. Second, we use standard tools in the field to scan through large protein databases. And these tools will try and identify sequences that seem to be evolutionarily related to the target. And then those get aligned to the target sequence, and it produces something called a multiple sequence alignment, or MSA. Then we use other standard tools in the field to search through structure databases, and that will look through all of the experimentally determined structures and find any that it thinks might be similar to the input. So that gives us our 3 inputs: amino acid sequence, the MSA, and any templates. And those inputs are then used to initialize 2 arrays of numbers. One represents the MSA, and then the other one, the pair representation, represents all residue pairs in the target. Unlike previous methods, AlphaFold isn't actually forced to pay attention to the inputs that you give it. So all of our tools are kind of tuned for high recall. If a few unrelated sequences sneak into the MSA or some bad templates are pulled in, then the model is sophisticated enough to be able to ignore that data. Whereas some previous methods would actually try and thread the sequence through an existing template. It doesn't follow things quite as carefully as that. Next slide, please. So then the main part of the AlphaFold network is composed of 48 layers of an architecture that's called EvoFormer. And EvoFormer really builds on some pre-existing ideas in the field of natural language processing. So this would be things like machine translation of sentences. In particular, it builds on an architecture called the transformer and on ideas like self-attention, which I can talk a bit more about later if anyone's interested to go into the details. And basically, these layers of the model will repeatedly update the MSA and the pair representation, and it will allow each one to inform the other until gradually, at the end of this process, the pair representation will contain informative values about the relationship between residues. So for instance, it might have some indication of which residues are close together in 3D space. Next slide, please. And then looking at the outputs of the network, we have a module right at the end called the structure module, which directly produces the 3D coordinates based on this final MSA and pair representations. Now, interestingly, this doesn't treat the protein as a chain, which I always found very strange. Instead, it treats each amino acid as a separate, independent rigid body, and it predicts one rotation and translation to place each of them in space. And the model actually learns that amino acids should be joined together as a chain. That's a behavior that sort of emerges organically, but we don't enforce it. And then finally, there are some comparatively simple neural network building blocks that do things like predicting the angles of the side chains. So those are the things, again, that you see drawn as sticks that sort of hang off of each amino acid. And we also have simple networks that produce two confidence metrics. You can see one of them at the top there, shown as a graph, which is called PLDDT. That's one of our confidence metrics. And then we have another called PAE that comes out of the pair representation. That's the thing that you see shown as a sort of green and white graph. And those have proven really important to help biologists interpret the predictions. Next slide, please.
So confidence metrics is what's the meaning of that? I mean, just in simple words, what do they show?
So I actually have a slide on that in the next section. So if you don't mind,
then
I'll wait until I have the appropriate pictures up, and then I'll
Sure.
tell you a bit about them. Grant. Okay. So because I'm aware that this is, a physics institute, and many of you may be doing work that has nothing to do with proteins, it's a possibility. I've tried to ask myself what we learned from developing AlphaFold that might be applicable to other projects. So hopefully some of this is interesting, but it's just some general reflections on building this network. Next slide, please. So first of all, obviously a lot of trial and error goes into developing a neural network. I think it would be crazy to say that, any piece of research isn't like that. But there probably were some general design principles that the ML experts bore in mind during this whole process. One thing is that we have produced an end-to-end network. That's one that goes all the way from the inputs to the structure, rather than, say, predicting an intermediate, like which amino acids are in contact, and then putting those into a separate, non-learned search process. And end-to-end approaches are thought to be useful because the model gets feedback about the entire process. It knows what it is that we want to predict in the end, and so it can optimize for that output at every layer of the network. We also built in what's called inductive biases, which is just a piece of machine learning jargon that means we used our existing knowledge about protein folding and built some of that into the design of the network. So, for example, there is the fact that the chain structure is de-emphasized, and instead we favor looking at pairs of residues, because physically pairwise interactions between side chains are one of the things that's expected to be important in this system. There's also a step in the network that considers triples of amino acids, and it basically allows each of them to update each other. And we think that this maybe helps to produce a self-consistent output in the end, because of course the final coordinates have to obey things like the triangle inequality. So on this sort of very high level, there are some things that we've used from our pre-existing knowledge and then built into the design. Inductive biases are thought to be particularly important for problems that have limited data. So unlike, many other ML projects where they can be downloading huge text data sets or huge image data sets from the internet, we're limited by the experimental structures that exist. So for us, this was an important thing to consider. Next slide, please. Another thing we learned is the importance of confidence metrics. These really weren't a focus of our project initially. We just wanted to get the protein structure right as often as possible. But once biologists were actually using our predictions in real-world applications, it turned out to be very important that the model also outputs something to give them an idea of where it's confident and where it's not. We actually ended up designing 2 separate metrics specialized for different tasks. So the one on the left there, PLDDT, basically gives you a per residue number between 0 and 100. And that tells you how confident is the model in the local structure just around that residue. But that's only sufficient for telling you about locally how confident is it. So then we also had to come up with a pairwise confidence metric, which is the one that you see on the right there. And that's called predicted aligned error, PAE. And this is to tell you about pairwise confidence. So say for the 2 rigid sort of domains that are highlighted in blue and red there, is AlphaFold confident in their relative positions or not? Because it could be that that whole protein is very flexible and those 2 pieces could be moving around in space relative to each other. And this will basically tell you, does AlphaFold have a hypothesis about how those 2 pieces fit together? Now, does that answer the question on confidence metrics, Patrick?
So yes, very much. So this, a naive look at this picture tells that it doesn't look too confident, right? This kind of prediction in this particular case, because it's just a diagonal line, right? As you said, it's mostly local confidence.
Exactly.
Right?
Yes.
But since I'm not a specialist, maybe actually it looks good. So what do you think?
So I would expect this protein to maybe be something we call beads on a string, which means that the individual pieces are sort of solid objects, but then they're joined by flexible linkers. So it may be that it could be in all sorts of different configurations, and that's why the distant pieces aren't confident relative to each other. That would be one possible interpretation.
I see. Thanks.
Next slide, please, unless there are other questions. The third takeaway is that just developing a neural network is really only half the battle. A lot of thought also needs to be put into what you do with it afterwards. And for this project, we decided to put a lot of effort into making AlphaFold available. So that meant not just writing a paper, but also providing a web interface for people to generate new predictions. And we open sourced the code, which turned out to be really important to the people who develop software for experimental structure determination. A lot of them have actually picked that up and very quickly been able to integrate it into the tools that they make, which has been really surprising and exciting for us to see, because it means that the adoption has been much quicker within the community. Next slide, please. As part of this whole release process, we had the opportunity to work with existing experts in the proteins field. And that is a general approach that I would wholeheartedly recommend. Machine learning people are experts in machine learning. It's very important to actually work with people who have domain expertise as well. So we ended up producing a large database of protein structure predictions in partnership with the European Bioinformatics Institute. They've been involved in cataloging big data for biology for absolutely decades. They're very trusted within the community. And so they were able to advise us on things like how to display the results clearly so that the confidence metrics are emphasized, versioning, metadata. And with their help, we've been able to expand the prediction database from what it was initially, where it was mainly all human proteins plus selected model organisms. And then we expanded it to SwissProt, which is a set of highly studied proteins. And now the database covers most of UniProt. So we've grown to about 200 million structure predictions at this point. Next slide, please. The final interesting thing we learned from this experience is that people will take a model that you've developed and apply it in all sorts of interesting ways that you never expected. So we obviously intended AlphaFold to be a tool for single protein structure prediction, but it's been used for all sorts of other protein-related tasks. And as an example, you can take something like intrinsic disorder. It's actually the case that not all protein sequences will fold up into a 3D structure. Some of them remain kind of disordered and fulfill their function in that shape. And various people have studied the relationship between low confidence regions of an AlphaFold prediction and disorder. And they've come to the conclusion that low AlphaFold confidence can be useful as a disorder predictor. Of course, not every low confidence region should be interpreted this way. There will be some that are down to a lack of input data to make a good prediction. And of course, there will still be genuine misses from the model. But there is enough of a relationship there for it to be useful. And so we've seen AlphaFold being used for disorder prediction as well as structure prediction. Next slide, please. One quick note of caution on that. Obviously, if you're going to apply an existing machine learning model to an entirely new problem, it's important to sanity check that it still performs well. And so practically speaking, we encourage people to do benchmarking, which means putting together a new test set that's relevant to the problem, running the model on them, scoring the results, and then comparing how the model does relative to other strong methods that already existed. At least for AlphaFold, you should always test on proteins that were released in PDB after April 2018, because anything prior to that was in the model's training set. Luckily, it doesn't take vast resources to actually run a benchmark like this. I've found that even looking at, tens or, maybe 80 or so structure predictions, if they're reasonably well-chosen proteins, that can already be quite informative. And if you use our web interface, then you can run those predictions on a GPU in the cloud rather than necessarily on your own hardware. So hopefully, if groups are interested in exploring new applications for AlphaFold, that should be something that's reasonably accessible. Okay, next slide, please. So again, I'm aware that there may be some non-biologists in the audience today, but I hope you'll bear with me because I would like to show you a few highlights of how AlphaFold has been used in the life sciences so far. For me, this is the really exciting part of the talk because we're not just doing machine learning for the sake of it. We're hoping to have an impact on the research that people do. And that's why it's been incredibly motivating for the team to see some of these applications. We actually have, a thread of people posting interesting Twitter links and preprints and things that they've seen. We really do follow all of the things that people are doing with the method. Next slide, please. So first of all, most proteins obviously don't work in isolation. They work by getting together with a bunch of other proteins and forming into a complex. And in particular, some proteins assemble into these really big structures that are so-called molecular machines. An example would be something like the ribosome, which has many, many protein components. Studying these large complexes is a really hot topic right now. And it's typically done using cryo-electron microscopy. So you take many, many images of the complex that you're interested in, do some data processing on those, and you get out at the other end a density, which provides a sort of outline of the protein. And in some cases, that will already give you enough detail, actually, to see the full molecular structure. But in other cases, the resolution might be not so great in certain places. And so you get the rough shape, but you maybe don't have the full atomic detail of the protein. So one common way that people have been using AlphaFold is to predict the structures of the individual protein pieces inside a big molecular machine. And then they will fit those into the cryo-EM density, kind of like a jigsaw. And that can help to build complex structures. For one thing, it can make more complete structures, and it can also speed up the whole structure building process. Poster child for this approach is the nuclear pore complex, which you can see there. This is a molecular machine that has hundreds and hundreds of subunits, and it acts as a gateway in and out of the cell nucleus. So this year, 3 different groups all published more complete structures of the nuclear pore, Those went into a special issue of Science, and 2 out of 3 of those groups had used AlphaFold as one of the tools to build these models, which was super exciting. Next slide, please. This is actually becoming a really standard technique, and I wanted to highlight one other example because it's a fun story. So just a couple of days after the AlphaFold database was released, someone on our team saw this tweet from Ben Engel, pointing out that we had some promising looking predictions for several components of another big complex, which is called the intraflagellar train. So Ben was already thinking to himself, like a day in, we have all the pieces now, is it time to build the train? And as you can imagine, we were all speculating about whether this was actually going to happen. But just a couple of months ago, we saw this second tweet from Gaia Pagino, I think, which said that the structure of the IFT had indeed been solved, which was awesome. And the team gave some credit to AlphaFold for helping to get the job done, which was very nice. But what that group did is a really impressive piece of experimental work, and it's a beautiful structure. So if you're interested, I'd definitely encourage you to check out their preprint there. Next slide, please. So at this point, the field is going beyond just sort of slotting AlphaFold predictions into experimental data. We're starting to see people building on this with more advanced methods. So for instance, someone called Tom Terwilliger has this really nice process where you can iteratively refine a prediction. So you take an initial AlphaFold model, you fit it into the experimental data, and then you use that as a template to feed back into AlphaFold. And so it goes on, and you're able to refine the prediction and get something that agrees closely with the data. But it's not just in experimental structure determination that AlphaFold's being used. It's also useful in other experiment planning contexts. So for instance, the images at the bottom there, you can see there's a transporter or a channel really. And the researchers wanted to disrupt the pore that goes through the middle of it. So they can look at a prediction and see this is exactly where the channel is, and these are the residues we need to mutate. Next slide, please. So I think I'll start going through these a bit quickly, but another exciting application has been predicting protein complexes and protein-protein interactions. AlphaFold was really designed to be a single protein system, but within days of launch, people had developed these hacks for getting it to predict the structures of 2 proteins bound to each other. And essentially the way that they did this was just by joining the 2 sequences together with a short linker. And that turned out to immediately give quite good results, which was a surprise to everyone, I think. And so within a matter of months, people had used AlphaFold together with these tricks to predict protein complex structures on quite a large scale. This paper by Humphreys et al. Is probably the best known example. There they were focusing on sort of evolutionarily conserved structures from eukaryotes. And they managed to determine a whole bunch of new complex structures and publish them in this paper. Next slide, please. So since then, DeepMind has actually published a model that's designed from the ground up to predict protein structures as complexes. That's called AlphaFold Multimer. And to give you some idea, it was basically retrained on complexes with a focus on cropping out interfaces, because that's the region that we want the model to learn about. And it's slightly more complicated to score protein complex predictions, because if you have multiple identical pieces, then you can get multiple permutations that are all correct. So we have to try and take that into account in the reward that the model gets. But with all of these tricks, we were able to develop something that's even better at predicting protein complexes and outperforms the linker method that I described before. Next slide, please. So the final thing I want to talk about is structure search. Basically, structure used to be a rarity, but now we have these huge databases full of predictions. And so it's possible to use structure-aware algorithms to search those. And tools for doing that have already been developed, most notably, I think, FoldSeek, which is highlighted there. And This is still sort of at the proof of concept stage, but I think one of the possible applications people have discussed is mining large sequence databases and trying to identify potentially useful proteins in them. So there is a paper that has done sort of a little bit of proof of concept work trying to identify useful enzymes using AlphaFold as one of the inputs. Next slide, please. Probably the most notable application of structure search so far has actually been identifying completely unknown proteins observed in experimental data. This is kind of wild. So the experimental data on the left there is cryo-ET data. It's essentially like the cryo-EM I was showing before. And they've got in gray there the outlines of a whole bunch of proteins that they've observed experimentally. But suppose that you don't actually know what protein is that piece that's highlighted in magenta. You only have its outline. One of the things that people can do now is they could trace out what that structure. Oh, great. Now I have no lights in here. Today just gets better on the technical front, huh? Give me 1 second. No one.
Look.
Yeah. So as I was saying, if you didn't know the identity of this magenta protein, you can basically trace out its structure and just search it against a whole database of, say, all of the proteins that exist in mouse, because we have structure predictions for them. And then you take the top hits, compare it with the density, and say, yes, that looks like my protein. And we've heard of several examples of groups actually identifying new interactions this way. Next slide, please. All right, so I think I'll finish with this section just to leave some time for questions. But it's good that we got to it because although I don't usually comment on this topic for today's audience, I thought I'd try and say a few words about the relationship between AlphaFold and protein physics. Next slide, please. So to give you, I think it's fair to say that going into the whole structure prediction project, what the field expected was that protein physics was going to really be at the heart of the solution. So for example, some people imagined that we would develop a model that can predict the free energy of a particular arrangement of amino acids. And then this whole thing reduces to a search problem, right? So you're searching through the free energy landscape and aiming to get to the minimum or at least a sort of metastable state that would then represent the folded protein. Other people might have expected we'd develop something like molecular dynamics, but you know, efficient enough that you could fold a protein structure from scratch. So some sort of a simulator. But the way that AlphaFold works is a bit unexpected. It's not quite like either of those things. Instead, what we have is a network that's good at extracting complex relationships from evolutionarily related sequences, and which is trained on all these families of proteins we've observed so far. So physics doesn't enter into this in a really obvious way, which begs the question, what, if anything, does AlphaFold know about protein physics? Next slide, please. Now, I'm quite a cautious person, and so it's in my nature to want to, you know, give a pessimistic answer to that. I think for a long time I would have just said, well, AlphaFold isn't a physical model. But there has been some recent work in the field that started to change my mind a little, actually. In particular, this preprint by Roni and Ovchinnikov. They were interested in whether AlphaFold can be used as a scoring function. So if you present it with several potential structures for a protein, can it identify which one looks most physically plausible? And the way they studied this was by taking away most of the evolutionary information, so no MSA, and then you feed in a potential structure as a template. AlphaFold will try and produce the best prediction it can based on that. And then you can develop a sort of composite score out of our confidence metrics, which tells you, you know, how plausible does AlphaFold find this structure. And what the authors found was that you actually can score structures quite well using this method. So if AlphaFold knows when the output is plausible and when it isn't, they speculated that it has at least internalized something about protein physics, and maybe even about energy functions, although you know, I think that's something that needs more study from looking at all of these structures that it's trained on. So that's one really interesting observation. Next slide, please. And then there are cases where you don't really have that much evolutionary information, and yet we get a reasonably strong prediction. So these are things like short peptides or de novo designed proteins that haven't evolved at all. And the fact that we can produce reasonable predictions for these is also, a really interesting observation and suggests there might be more going on there. Next slide, please. So I think I'll finish here just because of the delays and so on, but I would obviously not say that protein physics is solved. There are so many remaining challenges in that area. And in particular, one thing that we haven't really touched on is protein dynamics. Proteins are constantly able to change their shape between multiple states. Often that's critical to their function. And we don't yet have models that can reliably output all of the different states that exist, right? We're predicting one static structure. And let alone, we can't predict how much time is spent in each of these states, or how the transitions happen from one to the other. There's also the important aspect of the effects of mutations. Sometimes it's less the case that a mutation slightly changes a structure and more that it completely destabilizes it, or it changes the amount of time spent in one state versus another. And there can be cases where that's really important, like with the SARS-CoV-2 spike protein, for instance. It binds to the receptor on human cells when it's in a particular position with the RBD up. And if you have a variant that changes how much time it's spending in the up position versus the down, obviously that's something we want to know about, but it's not something that can be addressed with current models. So this is something that we can expect to be a real challenge going forward. But I think it's at least encouraging that we've managed to get to the stage where we have models like AlphaFold that seem to understand something about protein physics, maybe about protein structure. And this should be really encouraging that we can then go ahead and take on some of these remaining challenges. I think I'll stop there just for the sake of getting some questions in.
Okay, thanks a lot, Kathryn, for the very nice talk. I think now we can take questions, and maybe I will start. So one of it's developing it's more computer power, or the algorithms are getting better and better, and how about the training? How long it takes, and you need some huge computer. Yeah, this is more technical.
So in terms of the training, it's actually fairly modest compared to many state-of-the-art machine learning algorithms. The real crazy sort of models in terms of needing a lot of computer, things like large language models, things that are trained on large portions of the internet. Those really require an awful lot of compute. Unfortunately, I can't remember the exact numbers offhand, so I probably shouldn't give you a number of GPUs, but it's not too crazy. And I would say that in terms of time, it takes about 2 weeks to train a new model. But that's if you want to get it all the way up to full accuracy. It actually makes most of the progress in like the first 15% or so of the time. So it really depends on where you want to end training in terms of how long it takes. I would say that a lot more of it was about algorithm development rather than just throwing more compute at the problem. If you look at some of the algorithms that we had in CASP 13, those completely went out of the window and were rewritten for this in a way that, as I say, takes more into account things like inductive biases. So I think that for a problem like this, where you're data limited, you can't just rely on compute. You have to think a bit more about the design of the architecture as well.
Okay, thanks. So Dimitri, please.
So let me pass the word to Konrad, because I'm the organizer. So I want to give the opportunity for the participants, and also Konrad is from the field, and certainly he will ask a more smart question than I will.
Hello, you're on mute. Sorry.
Go ahead, Konrad.
Okay. I was wondering what happens if you give with disordered, partially disordered proteins. Do you get nonsense out? Do you get a flag out saying, I don't know what's going on here? Do you get the folded parts correctly and you shouldn't? How do you know? Maybe put it differently. Is there anything like an uncertainty estimation on the output?
Yep, absolutely. So for something like disordered regions, we would look at the confidence metric that we call PLDT that comes out of the model. So that gives you a sort of uncertainty metric. And we think that regions with a PLDT below about 50 for our public models, that tends to be associated with disorder, at least for proteins where you have enough input data to make a prediction, and there's no other reason why it should be uncertain. So in some of those cases, we think that that's associated with disorder. But what you would see is like a ribbon-like structure, something that isn't folded up, but is more like a sort of spaghetti, is what a disordered protein looks like. And there's also a possibility that there may be more unusual bond angles and lengths because the model is overall less certain on that region.
Okay, thanks. And could you imagine that you could use some future version of AlphaFold to kind of detect if a protein will hard to say them. So basically, there are many disordered proteins are in fact don't stay disordered, but for their function become ordered by associating with some other molecule. And now, of course, this is getting into complex system ligands and all this kind of stuff. But is this something you're working on and for the future?
So I would say that in some cases that can actually already be done. So there are cases where people have folded a disordered protein together with its binding partner using AlphaFold multimer. And in some cases, the disordered region will sort of slot in to where it's supposed to go on the structured partner. And I have seen a couple of examples of people using that in papers for things like short linear motifs, slims. They would show where that binds on the partner. I think one of the limiting factors there is that you do need to know what the disordered protein binds to. And I imagine that in at least some cases, we don't even know that yet. And it becomes a huge search problem if you then wanted to run it against many, many things. But yeah, promising movement in that direction.
I think sometimes you know what the binding partner is, but it's not a protein. It's some other molecule. So then...
I see. Yeah.
Anyway, thank you very much.
Who's next?
So, Fabio, please.
Hi, Thiago. Thank you for the word. But Dimitri has raised his hand first. I can speak, Dimitri.
Go ahead, Fabio.
Okay, okay. Thank you, Kathryn. Thank you for the good seminar. Look, as far as I could follow you, in some moment when you presented the design principles, you marked some points in your investigations, and you said that it was better to focus on the pair representations than in the residue. But after, when you were studying the mutation, you said that the residue were more important. Why this change in the statements?
Okay, so I wouldn't say that necessarily just because individual residues are important in point mutations, that means that you have to arrange the architecture such that it is focusing on individual residues. The motivation for focusing on pairs was really just about the importance of pairwise interactions in folding. But having said that, at the moment, AlphaFold is not that sensitive to point mutations. So who knows, maybe we do need to redesign the architecture and rethink some of these things in order to solve that problem. But right now, that's a bit of an open question, how best to capture mutations.
Right. Thank you for your answer.
Okay, I guess I can ask my question too. So the first question is related to Konrad's question. So if you, let's say, trying to predict some membrane protein, or well, as compared to a solution protein, is there any difference in how you do this?
So the actual process is exactly the same because the model isn't membrane aware. So it doesn't know any difference between the two of them. And although there are probably fewer training examples of membrane proteins in the PDB, There were some of these targets in CASP, and the model seems to cope with them pretty well. The only caveat I will say on membrane proteins is that, especially for single pass ones, sometimes the model will sort of distort the structure, so it ends up bent around like this. And we don't know fully why it does that yet, but because it doesn't have any awareness of the membrane, obviously it might end up putting the two ends closer to each other than they should be. You'll be able to tell that from looking at the confidence metrics. So it won't be confidently asserting that the start and the finish are close to each other. It's just they've ended up that way because the middle helix is distorted.
I see. Yeah, so you explained actually, so initially there is no physics that entered in the model, but maybe I have this comment that if you are so good in predicting the structures, so you can really accept it as a reality, which sometimes is better than the experimental results, and try to test your physical models just using, it's just much cheaper and quicker way to test. No, well, energy function is one example, or so in this relation, yeah, I don't know if you have any comment about this, but
so I think everything that I wanted to say on the relationship to physics is basically summed up as, you know, we think the model might have internalized something from all of the proteins that it's been trained on, and it certainly seems to be able to make some predictions that we find surprising. If it were just relying on evolutionary data, then it's hard to know where these results would be coming from. So there is a possibility that it might have some applications in physics. But honestly, I would always encourage people to benchmark, as I described earlier. You know, if you have some new application and you want to find out how well it does at this new task, then the key is just to test it out and find out, rather than for sure relying on my intuitions about what AlphaFold knows and what it doesn't. So some encouraging signs on physics, but I think we still have quite some way to go there.
So in this respect, you said that the code and the data are available. So can I just go and enter, you know, my random sequence of amino acids there, and then I'll get something from the algorithm? Or what are,
yeah, absolutely.
And what are the chances to get something sensible?
So that's obviously going to depend on the input, to be honest with you.
Right.
There are various factors that go into how likely AlphaFold is to make a good prediction. I would say that the most major one is how many evolutionarily related sequences it can find in the standard databases. So for some species, we just don't sequence very many things that are related to them. They're quite obscure organisms. And so that can be an issue. But in terms of how you get predictions, yes, there is a web interface. It's something called a Colab, where you can just input your sequence and then hit run, and you'll eventually get back a prediction run on our servers. So I normally go into that a bunch more, but I wasn't sure if people would actually be interested here.
It sounds like lots of things to do that physicists can do, actually. Yeah, so this one question that I have related to one of my favorite subjects, which are knots, and I know that people study knots in proteins. So can you predict knots with AlphaFold?
So AlphaFold will sometimes output knots, but I wouldn't necessarily trust those. I would say that in most cases, a knot would probably result from a low confidence region where the model, you know, just has to place the residues somewhere. It would usually place them in a sort of long arc like this, but there may be some cases where it just gets wrapped around itself. And in most cases, I wouldn't really set much store by that prediction. Having said that, you know, I've never actually benchmarked proteins that are known to form knots. So it's an investigation you could do. Like if you have your favorite examples of knotted proteins, just feed them into AlphaFold and see what it produces. Obviously bearing in mind whether they're in the training set or not.
Okay, well, thank you very much for this beautiful talk.
Okay, I think I have one more question. Before the talk, we are talking about AlphaGo and AlphaZero, and you mentioned what is exactly the difference? It's a different algorithm, different. Are there many different ways of approaching this machine learning, and what? If you just can comment on.
Yeah, absolutely. So in terms of the earlier work on games, so things like chess and Go, those are really a sort of search problem. So what the model needs to do is it needs to look several moves ahead, and it needs to figure out if I take this particular action, is it going to get me into a good position or a bad position? So the model needs to be able to search, and it needs to be able to evaluate how good a particular position is. And so there are separate machine learning techniques that you use to develop a network like that. Whereas the problem is slightly less of a search problem, I guess, with AlphaFold, because you have a bunch of training data where you already know the structure that you need to get to in the end. And you're essentially supervising the network and saying, okay, adjust your parameters so that the output prediction is closer to what I know the correct answer should be. So they're just slightly different setups in terms of how the problem is framed. And because of that, they do use quite different techniques. But that's not to say that we didn't learn anything from the previous work. I would say that mostly what we learned from things like AlphaGo and chess and so on was organizational things around, how to organize a team, how to keep everyone focused. All of that prior experience did feed in, but it was more in terms of how the company organized things rather than the actual machine learning methods.
Okay, so I'm not seeing any further questions. So thanks a lot for the beautiful talk, and thanks everybody for participating, and see you next week with more talks.
Thank you for
Thanks.
organizing.
Thank you.
Thank you very much.
See you guys. All the best.
Bye.